And it is really the story of how we make glucose. And so let me show you what we have on this poster. First, this is a chloroplast inside of a plant cell. These green stacks are called thylakoids. The area that surrounds the thylakoids is called the stroma. The light reaction happens on the thylakoids. The Calvin cycle happens in the gel that surrounds those thylakoids. It's called the stroma. We know that plants need water. We're going to show water coming in over here. We know that plants need carbon dioxide. That's going to be coming in right here. And I think that's it. Plants need some light energy, and that's the first step of this light reaction. The light energy comes in and it hits these thylakoids. Sitting on the surface of the thylakoids are electrons, and when that energy hits these thylakoids, the electrons get very excited and they fly outside of their usual orbits. And after they go up, they come down. And when they are falling, we are able to do some work with them. So the first thing we do with the falling electrons is we have them take those water molecules and they break the water molecules apart. They break the H's and the O's apart from each other. Now I have all these H's and O's and I have to know what to do with them. Well, the oxygen is what plants get rid of. That's their garbage. So we get rid of the oxygen. The H's are picked up by an H acceptor that is called NADP. Here it sits. NADP comes by here, picks up an H, and it becomes NADPH. NADP has room for one H, it picks up an H, it becomes NADPH. And we just keep breaking more and more water molecules until eventually we have 12 H's that we have saved, 12 hydrogens. And so I'm going to show these hydrogens right here. Here's 6 of them. Here's 6 more of them that I'm going to use later in the Calvin cycle. But that's what these red beads represent, is just hydrogen atoms that will be used later on. So oxygen I got rid of. My hydrogens I'm saving on my NADPH H acceptor right there. All right, the other thing I need to do in the light reaction is I use the energy from those falling electrons to charge up some ATPs. And in my model here, I've got adenosine diphosphate, which just has 2 phosphates, and I'm going to use the falling electrons to put one more phosphate onto ADP, making it into ATP. So there's a charged battery, a little unit of energy that this is going to be able to use to put together our sugar. So here's an ATP, here's adenosine diphosphate becomes adenosine triphosphate, and the energy for putting that little ATP together comes from those falling electrons. So I've got these charged batteries, and I'm just going to keep doing this in the light reaction until I have 18 ATPs of energy. I've only shown you 2, but we have to know that we've got 18 ATPs of energy. So I'm going to set those aside because I'm not going to use that model anymore. In the Calvin cycle, I'll show you how those ATPs get used. So here begins the Calvin cycle. In the Calvin cycle, we have to go through 2 times to make glucose. This is how it happens. I've got a 5-carbon chain that's sitting there, and we're going to call that 5-carbon chain Rubisco. And what the Rubisco does is it keeps being recycled and recycled in the Calvin cycle. And here's how it goes. First of all, the Rubisco comes by and picks up one more carbon from carbon dioxide, and it becomes a 6-carbon chain. Rubisco is a 5-carbon chain. It picks up one more carbon from carbon dioxide. Rubisco picks up one more carbon from carbon dioxide, and now I have 3 chains of 6 carbons each. Now those 3 chains continue around the Calvin cycle, and when they get to this spot, energy from ATPs is used to break those chains in half. So I just used 6 ATPs of energy. Once an ATP is used, it becomes an ADP. It goes back to the light reaction, gets another P put on, it becomes ATP again, and this is where we use those ATPs, breaking apart, rearranging our carbon chains. Now, I've got these 6 little 3-carbon chains and hydrogens. 6 hydrogens that we collected from the broken water in the light reaction are going to attach to one of those 3-carbon chains, and it's going to go out of the Calvin cycle. And you might recognize this, it's half of a glucose. But anyhow, 5 3-carbon chains continue on around. When they get to this spot, this is gonna remind us that we use some more ATP energy. 3 ATPs of energy are used to rearrange those chains into 3 chains of 5 carbons each. And now we're back to our rubisco. That's our starting point. The rubisco each picks up 1 more carbon from carbon dioxide. We've got lots of carbon here. Each rubisco binds with 1 more carbon from carbon dioxide. And now I've got 3 chains of 6 carbons each. 6 ATPs of energy. Break those chains in half. Now I have 6 chains of 3 carbons each, and my hydrogen that I collected from the broken water molecules binds to 1 of those chains. It goes out of the cycle, but 5 3-carbon chains continue around. I finally use 3 ATPs of energy to put these chains back into rubisco, a chain of 5 carbons. That rubisco just keeps getting recycled over and over, and every time it goes through the Calvin cycle, half a sugar comes out, half a sugar comes out, half a sugar comes out. These are going to be put together with a little dehydration reaction to form glucose. And you can see from this poster how very exciting that conclusion is.